Although there's not time to go in depth here into amino acid and lipid metabolism, my goal for this video is to introduce you to the main concepts, the key regulatory points, where things are occurring and why. Let's start with amino acids. If you think about amino acids, we have some variety, but all of them are going to have at least one amino group, and then they have some sort of carbon skeleton. That carbon skeleton is often very useful. We can break it down, we can oxidize it to get ATP. We can interconvert it with other molecules to make other molecules, various things, but all of those are going to require removing this amino group. We can remove this amino group via a process called transamination, we'll talk about, and that's going to be the first step to being able to use the carbon skeleton. Now that we have that carbon skeleton, we can go use it, and the fate is going to depend on the identity of that carbon skeleton. So if you have something like pyruvate left over versus if you have something like alpha-ketoglutarate, those will have different purposes that they can be used for. But what are you going to do with that amino group? The amino group, we can't just chop it off because that would give us ammonia, which is toxic. So instead, those amino groups are going to be disposed of safely as a urea via the urea cycle, which takes place in the liver. The urea cycle takes place in the liver, but we're going to be breaking down proteins and amino acids in all sorts of cells. And so we'll see how we can transport these amino groups safely through the bloodstream to the liver, often in the form of alanine or glutamine. So our first step is removing that amino group. And again, we're not just going to chop it off typically because that would give us ammonia. We're going to instead transfer it. When we transfer an amino group off of an amino acid, what we're left with is an alpha-keto acid. So now we have a ketone at this alpha carbon rather than having an amino group at the alpha carbon. We can pass one amino group from an amino acid onto an alpha-keto acid to get another amino acid and be left over with an alpha-keto acid. Often what we're going to be doing is we're going to be transferring an amino group from some amino acid onto alpha-ketoglutarate, and that gives us the amino acid glutamate. So glutamate is going to be your first stop for most of the amino acid transaminations that we'll see. And this kind of simplifies things so that your cells can now deal with glutamate as opposed to having to deal with each of the 20 amino acids separately. The enzymes that catalyze this transamination are logically called transaminases, and they often use the cofactor called pyridoxal phosphate, or PLP. These reactions are typically readily reversible, and so they can go in either direction. You can do a lot of easy conversions between alpha keto acids and alpha amino acids. Depending on which amino acid you are transaminating, you're going to be left with different carbon skeletons. So you can take an amino group from any amino acid, stick it onto alpha-ketoglutarate, and that'll give you glutamate. But the alpha-keto acid you're left over with from that amino acid you remove the amino group from is going to vary. A couple of the main pairs, you can take alanine and you can get pyruvate left over. You can take glutamate, you get alpha-ketoglutarate left over, and you take aspartate and you get oxaloacetate left over. These are key pairs because if we think about it, well, glutamate, we get alpha-ketoglutarate. Alpha-ketoglutarate is part of the citric acid cycle. Aspartate can interconvert with oxaloacetate. Oxaloacetate is part of the citric acid cycle. And alanine, well, alanine, we transaminate it, we get pyruvate. Pyruvate can be oxidatively decarboxylated to give us acetyl-CoA, which can then enter the TCA. Or it can be transformed into glucose. It can be transformed into lactate, all sorts of various things that you could do with that pyruvate. So those are kind of three of the main pairs, and they're especially helpful because they can be done in one-step transformations. Other amino acids can be broken down into things that are either in the TCA or things that are adjacent to the TCA, various different carbon skeletons, but sometimes those are going to require multiple steps. The amino acids that can be broken down into components of the citric acid cycle or pyruvate, we refer to as glucogenic. Whereas amino acids are classified as ketogenic if they break down into products that are either acetyl-CoA or acetylacetyl-CoA. Why this distinction? This goes back to thinking about anaplerotic reactions, which are reactions that regenerate pathway intermediates. The way that we can make glucose is by removing malates, which can be transformed into oxaloacetate, which can be used for gluconeogenesis. Note that it's oxaloacetate that is in the gluconeogenesis pathway, but malate can be removed from the mitochondria, whereas oxaloacetate cannot. If you're breaking down an amino acid into something that is already in the citric acid cycle, then you can remove something from the citric acid cycle. You could remove that oxaloacetate, or you can remove that malate, and you'd still be able to keep the cycle going. And so you could use this in order to sustainably make glucose. Now imagine, however, that you were to take out that malate or you were to take out that oxaloacetate, but all you were adding in was going to be an acetyl-CoA. Well, you wouldn't even be able to add in the acetyl-CoA because you're taking away the oxaloacetate. You're not adding more carbons than you're taking away, and so you can't sustainably make glucose that way. But if you do have an amino acid that makes acetyl-CoA or makes acetyl-acetyl-CoA, those can be hooked up into lipids. They can be sent through the bloodstream as ketone bodies, typically as this beta-hydroxybutyrate, and so that's why we call them ketogenic. And some amino acids are both. Some amino acids are actually going to have multiple products. Some of those would be glucogenic, and some of them would be ketogenic. So if we remove those amino groups from amino acids, we can get a bunch of useful carbon skeletons. But what about that amino group? We saw how we could pass it on to alpha-ketoglutarate to give us glutamate. But we don't want to be taking away all our alpha-ketoglutarate, so we need a way to get that amino group back off of glutamate so that we can have more alpha-ketoglutarate, both to keep the TCA going, as well as to be able to remove more amino groups from other amino acids. How are we going to remove those amino groups? We're going to remove them through the urea cycle. The urea cycle is going to safely remove those amino groups as urea, rather than as ammonia. But there's a complication because the urea cycle is happening in the liver, but we want to be breaking down amino acids and breaking down proteins in all sorts of tissues. So how are we going to get those amino groups safely there? They're typically transported through the bloodstream in the form of glutamine or alanine. Now, I know we were just talking about glutamate, but glutamate is going to be charged and it's a neurotransmitter and things like this, so we don't want to just be sending it through the bloodstream. So instead, we are going to send glutamine, or we're going to send alanine. With glutamine too, now we're sending more amino groups than if we were just to send our glutamate. And so most of your tissues are going to be sending glutamine, but your muscles are going to be sending alanine. Just like we saw that there was the cycle between our muscles and our liver with the lactate, there's also a cycle between our muscles and our liver with glucose and alanine. The muscles, when they break down protein, they take an amino acid, they transaminate alpha-ketoglutarate to give you glutamate. Now they're going to take that glutamate and they're going to stick it on pyruvate to give you alanine. That alanine can then go and it can go travel through the bloodstream, and it'll travel through the bloodstream to your liver. In your liver, that amino group is going to be stuck back on alpha-ketoglutarate to give you glutamate. Now this is happening in your liver, so that glutamate can go and it can take those amino groups through the urea cycle and regenerate the alpha-ketoglutarate. When you remove that amino group from the alanine, well, now you're left with pyruvate. Pyruvate, you're in your liver, gluconeogenesis, ding, 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 ding. You can make glucose that you can send back to your muscles. Your muscles then go through glycolysis, they generate pyruvate, they break down proteins, they generate amino groups, they put those amino groups onto alpha-ketoglutarate to give you glutamate, and then they transfer those amino groups from the glutamate onto the pyruvate to give you alanine. By doing it in kind of like two steps that way, you're able to focus on using this glutamate as the transfer rather than having to think about using each amino acid separately to get onto pyruvate. So you have just the one that you need to focus on to get the amino group from glutamate onto the pyruvate. That gives you alanine. You send that through the bloodstream to the liver. The liver then takes that amino group, sticks it back on alpha-ketoglutarate to give you glutamate. The glutamate then takes those amino groups to the urea cycle to dispose of them safely as urea. The pyruvate that's left over gets made back into glucose, which gets sent back to your muscles. Once again, your muscles are making your liver do the hard work. So what's going on in the urea cycle? We're not really going to go into much of the details, and I have much more details and videos and things if you want to learn more. But the bottom line is that those amino groups that you transferred through the bloodstream in the form of that glutamine or alanine get into the liver. Those amino groups are now passed on to alpha-ketoglutarate to give you glutamate, our good old friend. Now those glutamates are going to be used in order to get the amino groups into this molecule of urea that can be excreted. Some of this takes place in the mitochondria, some of this takes place in the cytoplasm. Note that one of the glutamates is actually going to transfer its amino group onto oxaloacetate to give you aspartate. And so that's why if we looked at the TCA charts, we saw aspartate was being used in the urea cycle because aspartate is being used in the urea cycle, and so is glutamate. So you have multiple nitrogen groups, 2 per urea, that are actually going to be released. But by using this pass off from glutamate, again, you're able to then have this focus on the glutamate and be able to transfer amino groups from any amino acid, such as your glutamine or your alanine, onto alpha-ketoglutarate to give you glutamate. And then you can focus on using the glutamate to give you the aspartate or to give you that amino group, that is ammonia. Because you do have an ammonia that's generated, but that's in your mitochondria, and it's quickly captured in the form of this carbamoyl phosphate. The enzyme that does this capturing is called carbamoyl phosphate synthetase 1, CPS1. CPS1 is the regulatory point of the cycle. So the cycle is mostly regulated just based on quantities of the various components. So we've got our thermodynamic regulation. But carbamoyl phosphate synthetase 1, where you're capturing that ammonia, is going to be the key regulatory step. It's regulated indirectly through arginine levels through an allosteric regulator called N-acetylglutamate, or NAG. So this is the regulatory step of the urea cycle. And the purpose of the urea cycle is to remove those amino groups as urea rather than as ammonia, because we don't want Windex veins. As I mentioned, a lot of these reactions involving amino acids and their interconversions are going to be readily reversible. So just like we could take amino acids and put their skeleton pieces into the citric acid cycle, we can also use skeleton pieces from the citric acid cycle and from things you can get from other compounds in order to make amino acids. We need all of our amino acids. However, only some of them are considered essential. All that means is essential in the dietary sense, meaning that our bodies can't make them from scratch ourselves, and so we need to get them pre-made through our diet. So that's all that's meant by essential. We can't make them from scratch, but we still need all of the amino acids. Some of them, the most important ones, are often the ones that we can make ourselves. We need to have that machinery to make them ourselves because they're so important. And some amino acids are what we consider conditionally essential because we can make them from essential ones. We could make them, but not fast enough to keep up at some stages of our life and things like this. So the key points about amino acid metabolism, and then we'll move on to lipids. There are 3 main steps of amino acid metabolism. You remove the amino group, typically via transamination, which are typically going to be those easily reversible reactions. And typically we're going to be sticking an amino group onto alpha-ketoglutarate to give us glutamate, and then just dealing with the glutamate. Now we're left over with an alpha-keto acid skeleton. Depending on the identity of that skeleton, we can do different things. And some of these can be considered glucogenic if they are broken down into pyruvate or something that's in the citric acid cycle. And if that pyruvate counts, but not acetyl-CoA, even though neither of those are in the TCA already, because pyruvate can go through that anaplerotic reaction through pyruvate carboxylase, that's going to give you oxaloacetate, which is in the cycle. But acetyl-CoA, it only has 2 carbons, and it can't enter the citric acid cycle unless the cycle has oxaloacetate to join up with. Pyruvate could give you oxaloacetate, so it was going to be glucogenic. But acetyl-CoA can't, so it isn't going to be glucogenic. Instead, it's ketogenic. Now we're building up that glutamate, though, and so how are we going to actually get rid of the amino groups? We're going to get rid of those amino groups as urea via the urea cycle in the liver, which is going to prevent us from having to have a bunch of ammonia building up. We're going to get those amino groups to the liver in the form of glutamine or alanine. And then the liver is going to take the urea cycle, remove those amino groups safely for us. Now let's move on to fats, lipids, and fatty acids mainly. First, let's orient ourselves. Let's think about where things are happening. All of our cells are going to need fatty acids, but not all of our cells are going to have the capacity to make fatty acids. Your liver and your fat cells are going to be the main cells that are going to be able to make fatty acids. They make fatty acids in their cytosol in a process that uses NADPH. And remember that the pentose phosphate pathway was the main source of our NADPH. So our pentose phosphate pathway is going to be providing the NADPH that we can then use in order to synthesize fatty acids. And that synthesis is happening in the cytosol of our liver and of our fat cells. So our hepatocytes and our adipocytes. Your liver and your fat cells are going to store those fatty acids in the form of triacylglycerols. So stick 3 of them onto a glycerol molecule. That's great. Now you've got this good long-term storage form. But in order to use it, you want to actually be able to remove the fatty acids. And so enzymes called lipases will chop the fatty acids off of the glycerol backbone to give you free fatty acids. Fatty acids are delivered to other tissues from your liver in the form of actually tags in lipoproteins, or from your fat cells as fatty acids piggybacking on serum proteins. Other tissues can then incorporate those fatty acids into more complex lipids in their ER. And then they can also break down those fatty acids for energy. That breakdown occurs in the mitochondria in a process called beta-oxidation. So your cells throughout your body are doing beta-oxidation to break down fatty acids. But only your fat and your liver cells are going to be making fatty acids. They're going to be making them in their cytosol. Then they're going to be sending fatty acids or triacylglycerols to various tissues. The tissues can then use them for various purposes, making more complex lipids, or they can break them down. That breaking down happens in a process called beta-oxidation that happens in the mitochondria. Fatty acids are made from acetyl-CoA, and they're broken down into acetyl-CoA. This making and breakdown are going to be inversely regulated, so you're not doing both at the same time. In a little more detail, remember that our liver and our fat cells are going to be storing the triacyl. In a little more detail, your liver and your adipose tissue, so your fat cells, are making those fatty acids. They store those fatty acids for long-term storage in the form of triacylglycerols or tags, which is the same as a triglyceride. This is great for storage because now you've got this really hydrophobic thing. It's not going to be keeping a bunch of water hooked up with it. And so you've got this long-term storage. In order to be used, however, you've got to free the fatty acids, and lipases are going to go and they can chop off the fatty acids using water, leaving you with a glycerol backbone and a free fatty acid. This cleaving can happen inside of the tissue, or it can happen extracellularly. Your fat cells are actually going to chop off the fatty acids inside of the fat cells and then go and ship them out, piggybacking on other proteins. Whereas your liver is going to send out these triacylglycerols, and then they're going to be removed from the triacylglycerols extracellularly. So we'll get more into this in a second. Your liver are going to be sending out triacylglycerols, so these tags. These tags can't travel just as is, free floating through the bloodstream, but they can be transported in the form of these lipoproteins. These lipoproteins are kind of like these bubbles that contain cholesterol and phospholipids and triglycerides and these apolipoproteins. So basically, you've got these bubbles of fatty stuff, but these bubbles have these phospholipids. So you've got a membrane bubble, not just having fat kind of clogging up your bloodstream. These bubbles get various names depending on how big and how dense they are. And so your liver is going to send out this very low density lipoprotein. So these big bubbles with low density, and then those are going to get taken throughout your blood through different tissues. And there's this extracellular lipase called lipoprotein lipase that's going to be able to chop off the fatty acids that are present in those lipoproteins. And this is going to allow the cells to take in the fatty acids. So the cells can't take in the tags, but they can take in the fatty acids. The Liver sends out the tags in these lipoproteins. Lipoprotein lipase on the surface of blood vessels is going to actually remove them. That's going to leave you with glycerol and the fatty acid that can be taken into the cell. This is going to leave that bubble smaller, and so then you end up with your higher density lipoproteins, and then those get taken back to the liver, and then the liver can go and it can add more tags and things like this. So it's a kind of complicated thing, and we're not going to go into that. So your liver is sending out those tags in the form of the lipoproteins. Your adipocytes, your fat cells, are actually going to send out fatty acids. So they do the cleavage off, the lipase step inside of the fat cells, and then they send the fatty acids to the body and to the liver to put into the lipoproteins. Inside of your fat cells, they're going to regulate the breakdown of their triacylglycerols. What they have is they have a hormone-sensitive lipase that is going to break down those fats in response to glucagon. Remember, glucagon saying glucose is gone. So if glucose is gone, we're going to need to get some energy mobilization going on. And that is going to make it so that you're actually going to break down those fats to get the fatty acids. Now, how are you going to get those fatty acids throughout the body? You can't just put those fatty acids free floating. You don't have a membrane, you just have a fatty acid. What are you going to do? And so you're going to stick that fatty acid, you're going to glob it onto a plasma carrier protein, such as serum albumin, that have these hydrophobic binding patches that are going to bind onto that fatty acid, allow them to be transported throughout the bloodstream. And now the cells can just take those up. A little more about this hormone-sensitive lipase, which remember is removing the fatty acids from tags inside of the fat cells. We mentioned how it would respond to glucagon. It can also respond to adrenaline, so you can really get the fuel throughout your body when you need it. Don't confuse this with lipoprotein lipase. Remember, lipoprotein lipase is going to be acting extracellularly to get fatty acids into cells from tags, such as the tags that were sent in those lipoproteins from the liver. How's this going to work is the glucagon and adrenaline are going to activate this through a GPCR pathway. That one that we've seen before where we're activating pKa in response to glucagon or adrenaline binding to the receptor. What that is going to do is it's going to make the pKa active. pKa is a kinase. It's going to then phosphorylate this hormone-sensitive lipase, which is then going to cause that hormone-sensitive lipase to go to the surface of lipid droplets and chop off the fatty acids to be used. So that's great if you've got glucagon or adrenaline telling you that you need to release some energy. But what if you got the opposite situation? You got insulin. Insulin is saying we've got a lot of glucose, let some in, and let's actually go and store it. And so if we're storing these as fats, we don't want to be breaking down the fats at the same time. And so insulin is going to activate a protein phosphatase. Remember, a phosphatase removes phosphate groups. So the phosphatase is going to remove the phosphate group that was put onto the hormone-sensitive lipase by protein kinase A. Protein kinase A was activated by glucagon and adrenaline, remember? And so here, we are reversing that actions of those hormones with insulin. It's going to remove that phosphate group. It's going to then remove the hormone-sensitive lipase from the surface of those lipid droplets. And so the fatty acids won't be released from those tags. So your liver takes those fatty acids and esterifies them. It takes those 3 fatty acids and sticks them onto glycerol to give you a triacylglycerol tag. It's great for long-term storage, but now you want to get it to other tissues. Your liver is going to package it up in the form of these very large density lipoproteins and send them throughout the body. On the surface of the blood vessels are lipoprotein lipase that can chop off the fatty acids from those tags, allowing free fatty acids to enter cells, where the cells can then use them. In your fat cells, your adipocytes, you're also going to be making triacylglycerols and storing them. But rather than sending them out as triacylglycerols for other tissues to use, you're going to send them out as fatty acids. So you've got to do the lipase step inside of the cells, and the hormone-sensitive lipase is going to allow you to do this in response to glucagon or adrenaline, free up those fatty acids to be sent throughout the blood. They can't travel just freely throughout the blood. Instead, they're going to piggyback on plasma carrier proteins such as serum albumin. Your intestines can also send out fat in the form of these chylomicrons that can then be taken up by, say, the liver to process. Now let's think about those fatty acids are actually made and how they're broken down. So we want to focus on this corner of our metabolic map mostly. Starting with the synthesis, fatty acids are built by a multifunctional protein called fatty acid synthase, or FAS. It's built from acetyl-CoA, but it's actually built indirectly through a 3-carbon compound called malonyl-CoA. And so you don't need to know the details. The bottom line is that you're taking acetyl-CoA, you're actually adding a 3rd carbon group from CO2. The purpose of this is to activate that acetyl-CoA so that it can be linked up. That 3rd carbon group that you added is actually just going to get removed. It's not going to be incorporated into the fatty acid. So you're only adding 2 carbon groups at a time, and this is going to cost you 1 ATP and 2 NADPH. And remember, the NADPH, we get that mainly from the pentose phosphate pathway. So we're going to be spending ATP and spending NADPH. Remember, NADPH we use for these reductive reactions. We're reducing these carbons, therefore we're kind of storing the energy for later. You do this in cycles, and each cycle you're going to be adding 2 carbons until you get to 16 carbons. A saturated 16-carbon fatty acid is called palmitate, and so you end up with palmitate by default. And then you can introduce things like double bonds, or you could elongate it in other compartments in your cell. The key regulatory step of this fatty acid synthesis process is acetyl-CoA carboxylase, or ACC. It's the enzyme that actually activates that acetyl-CoA to make that malonyl-CoA, that 3-carbon compound. And remember that the purpose of this was just to make it so that it was activated. What it does is it makes a beta-keto acid, which is energetically favorable to subsequently decarboxylate. And so that's why you have this. And it's so important that you're actually spending energy to do this. So this is where you spend the ATP. This is going to be your regulatory step, your key regulatory step. For each 2 carbons that you add, you're costing 1 ATP and then 2 NADPH. You do this in cycles until you get to that 16-carbon saturated fatty acid, or palmitate. Note that there's also the setup phase where basically you have to attach the first acetyl group to the fatty acid synthase and shift things over. But that is not going to go through malonyl-CoA. Don't worry about this. Just know this part, and don't worry about the details. There's a complication, however, because we see an acetyl-CoA could be produced in the mitochondria. We saw that it could be produced from the oxidative decarboxylation of pyruvate. However, remember, the mitochondria is really, really picky. And so picky that it won't let acetyl-CoA out. And so instead of letting acetyl-CoA out, we've got to sneak acetyl-CoA out, basically, and we're going to sneak it out in the form of citrate. So we can take acetyl-CoA, combine it with oxaloacetate to get citrate, ship citrate out of the mitochondria, and then convert it to acetyl-CoA, which we can then use. The citrate lyase step is going to be what makes the acetyl-CoA from citrate. It basically chops off the oxaloacetate, and you're going to have to spend an ATP in order to do this. The citrate lyase step is going to be important for allowing us to get the acetyl-CoA equivalent out of the mitochondria and into the cytoplasm, because the cytoplasm is where we use fatty acid synthesis. This is just one example of how we can sneak things in and out of the mitochondria. We saw with the electron shuttles, we can basically sneak electrons from NADH out through malate aspartate shuttle. We saw how we can use malate dehydrogenase to sneak oxaloacetate basically out of the mitochondria in the form of malate. And so here we're seeing that we can sneak the equivalent of acetyl-CoA out of the mitochondria in the form of citrate. Note here we have our acetyl-CoA carboxylase, or ACC, which remember was going to be the step that actually gave us malonyl-CoA, and that was going to be our very highly regulated step. We have this acetyl-CoA carboxylase, which is going to carboxylate the acetyl-CoA to give us malonyl-CoA. It's going to cost ATP, and it's going to use CO2 or bicarbonate HCO3- in order to add on those carbon groups. So we often use those terms H3 bicarbonate and CO2 interchangeably, so you could write it either way basically. but more accurate would be this HCO3 minus. This step is highly regulated, as we can see, and it's going to be activated by citrate. And so that's feed-forward stimulation and insulin. And so remember, insulin is saying, we've got a lot of glucose, let's let it in. We're going to stimulate this pathway. We want to stimulate the synthesis. We saw how we could inhibit the removal of the fatty acids from tags via insulin signaling. And here we're seeing that insulin signaling is going to actually promote us making more fatty acids from the glucose that we're taking in. Just how we saw the glucagon was going to inhibit us from storing the fatty acids, it's also going to inhibit us from making fatty acids. It's also inhibited by palmitoyl-CoA. Now, palmitoyl-CoA is something that you are going to be seeing if you are making fatty acids. So palmitoyl-CoA is going to be feedback inhibiting this process. And note that palmitoyl-CoA is the CoA attached version of palmitate. So let's talk about the CoA forms of fatty acids, because that is going to get us into fatty acid catabolism. So the fatty acid breakdown is going to occur in most tissues and take place mostly in the mitochondria in a process called beta-oxidation. If you have really long ones, those are going to be initially broken down via hydrogen peroxide mediated process in little membrane compartments called peroxisomes. But we're just going to focus on beta-oxidation. The basic process is that fatty acids are going to be snuck in. Another thing that we've got to sneak in, we'll talk more in a minute, but they're going to be snuck in via the carnitine shuttle and then broken down 2 carbons at a time. So we built them 2 carbons at a time, we're breaking them down 2 carbons at a time. So we built them from acetyl-CoA through that malonyl-CoA intermediate, and we're going to break them down into acetyl-CoA. And we're going to be doing this in cycles again. This time, our cycles are going to be generating NADH and FADH2, because now we're doing oxidation, we're generating NADH, not doing reduction, in which we're using NADPH. We go through these cycles. Each cycle, we're cutting off 2 carbons at a time, and the 2 carbons that we're cutting off, we basically cut them off in the form of acetyl-CoA, produce 1 NADH and 1 FADH2, which then we can use to take those through the electron transport chain and make ATP. So let's get into the details, but not too deep into the details, just the very basics. So beta oxidation is occurring in the mitochondria, breaking down 2 carbons at a time. And so if you start with a even chain fatty acid, you end up with just acetyl-CoAs. So you can break them down. So if you had your palmitate, say you've got 16 carbons, you break it down into 2s, and then what you're going to end up with is you're going to have 16 divided by 2 is 8 acetyl-CoA. And that would take you 7 cycles, because the last cycle is going to leave you with 2 CoA. Think about chopping an apple. You do 1 chop, you get 2 pieces. So you only need to do 7 cycles to break down into 8 acetyl-CoA. And then those acetyl-CoAs can actually go to the citric acid cycle. So not only are you getting the NADH and the FADH2 that you can take through the electron transport chain directly, you can also take the acetyl-CoA into the tricarboxylic acid cycle, assuming, of course, you've got oxaloacetate there. And now you're going to be able to go and oxidize that further, get more NADH, more FADH2, more ATP, all that energy. Woo woo! But remember that when you were making the fatty acids, you actually had to put in the ATP. Know, however, that we would not be able to sustainably make glucose from that, because we would need that oxaloacetate to be present. So this would not be glucogenic if we had an even chain fatty acid. But now let's imagine that we have an odd chain fatty acid. Now when we break it down, we're left with a 3-carbon piece. That 3-carbon piece is what we call propionyl-CoA. This propionyl-CoA can get converted to succinyl-CoA, and succinyl-CoA is part of the citric acid cycle. So if we start with an odd chain fatty acid, now we're making something that is already in the citric acid cycle, and so this would be glucogenic. So we can sustainably make glucose from those odd chain fatty acids, but not the even chain fatty acids. And I mention this mainly because it connects back to our thinking about amino acid metabolism. Again, we're seeing how we can convert all these bigger, more complicated compounds into these smaller pieces that we can then interconvert into other bigger, more complicated things. And so it really is logical. You just have to really kind of spend some time trying to get familiar with things. So this is just the process of making that succinyl-CoA from the propionyl-CoA, in case you were interested. What's kind of cool about it is that it's one of the only enzymes in your body that's actually going to use a cobalt. So now let's get to the complication of actually getting the fatty acids into the mitochondria to do the beta oxidation. When we do this breakdown, rather than breaking down a fatty acid, we're breaking down a fatty acyl-CoA. So our fatty acid already has a CoA attached to it. Then we're basically using CoA like a pair of scissors that gets stuck on what it cuts. And so we're cutting off 2 carbons, but keeping those carbons attached to a CoA. And so we need to have a CoA that's attached to the end in order to begin with. And so we start with a fatty acyl-CoA rather than starting with a fatty acid. But a fatty acyl-CoA cannot get into the mitochondria. So what we're going to do is we're going to use another one of our sneaky shuttles. This one is going to be called the carnitine shuttle. So carnitine is just like this little amino acid molecule. In the carnitine shuttle, we take a fatty acyl-CoA, and then we take carnitine and we put the fatty acid onto the carnitine instead of the CoA. We take that fatty acyl-carnitine into the mitochondria. Then we take off the carnitine, swap it back for a CoA. Now we've got a fatty acyl-CoA, which can go through beta-oxidation to give us that acetyl-CoA. In order to do this, you first need to make the fatty acyl-CoA in your cytoplasm. So in the membrane, the outer mitochondrial membrane, there's this fatty acyl-CoA synthetase, this ACS. And what it does is it basically attaches the CoA using ATP. And note that it's using ATP, but it's generating AMP and this PPI. So basically, it's the equivalent of using 2 ATP because you're going from ATP to AMP rather than ATP to ADP. So it's highly energetically expensive, but now we've got our fatty acyl-CoA. So we've got our fatty acyl-CoA, yay! But we can't actually use it right now. We can't get it into the mitochondria. Instead, first we've got to swap the CoA for a carnitine, take the fatty acyl-carnitine into the inner mitochondrial space. Then it's going to go through this translocase. This translocase is then going to take the fatty acyl-carnitine through. And now you're on the other side. So now we're inside the mitochondria, great. But we've got a fatty acyl-carnitine, which isn't going to be useful for our beta oxidation as is. So we need to reverse this process, the swap that we did out here. We need to reverse this, take the carnitine, and swap it back for a CoA to get our fatty acyl-CoA. The enzyme that's going to do this is going to be CAT2, carnitine acyltransferase 2. Thinking about regulation, this carnitine acyltransferase 1, CAT1, you might also see it called carnitine palmitoyl transferase. So remember, palmitate was kind of like our default 16-carbon saturated fatty acid. And so you might see it abbreviated CPT1 or CAT1. And it's going to be the key regulatory step of fatty acid catabolism. Because here you're kind of saying, okay, let's go actually go and take this into the mitochondria. So once you get to this acylcarnitine, then you can get into this inner membrane space, but you can't get into the mitochondria unless you go through this translocase. But the translocase, this is just going to be a passive transport, but you're going to be swapping it for a carnitine that you can then go take back to be able to do this again. So this functions as the cycle of swapping the carnitine in and out in order to get the fatty acyl carnitines in and out. Because what we want is going to be the fatty acyl-CoA, but we can't take the fatty acyl-CoA in there. So once again, we take our fatty acid, we put on a CoA using fatty acyl-CoA synthetase or ACS. And that's going to cost us the equivalent of 2 ATP. Now we've got this fatty acyl-CoA that we want to break down in the mitochondria, but we can't get it into the mitochondria. So we swap the CoA for a carnitine using this CAT1 enzyme. This CAT1 enzyme is going to be highly regulated because here we're saying, okay, let's go and take it into the mitochondria. We can now go and this can go through this carnitine acylcarnitine translocase into the mitochondria. And inside of the mitochondria, you have another copy of this carnitine acyltransferase, But this one is going to be Cat2. And so this is the inner mitochondrial version of it, as opposed to kind of like the cytosolic version of it. But both are going to be located in membranes. But what it's going to do is it's going to do the reverse process, remove the carnitine, put on a CoA, and now you got the fatty acyl CoA. You go through beta oxidation. Each step, you are cutting off an acetyl CoA. And so you're going to be generating the acetyl CoA, as well as NADH and FADH2. And now you can go use those for ATP making. To get into the TCA, however, you need to have oxaloacetate present. If you don't, what's going to happen is that you're going to build up acetyl-CoA. The built-up acetyl-CoA is actually going to join together with another acetyl-CoA. And so that last step of beta oxidation, that thiolase step, is actually going to reverse itself. We end up with acetoacetyl-CoA, which can be transformed into acetoacetate, which is one of our ketone bodies. It can spontaneously decarboxylate to give you acetone, which isn't great. And so instead, acetoacetate is going to get reduced to give us beta-hydroxybutyrate. And then this beta-hydroxybutyrate can be sent through the bloodstream to tissues like the brain that really need it. And so you can break down lipids in your liver. If you need to ship the energy from those lipids to the rest of the body, what you can do is you can send them as these ketone bodies. Too much of these, and what you get is ketoacidosis because you've got this acid. However, the ketone bodies are very important in times of starvation, so you don't have enough oxaloacetate. In cases of uncontrolled diabetes, when there's not enough oxaloacetate, heavy alcohol use, various cases, acetyl-CoA builds up, causes the formation of these ketone bodies. So to review some of the key points from lipid metabolism, our fatty acid synthesis is going to occur mostly in the cytoplasm of our liver and fat cells, and it's going to be a key user of NADPH, which, remember, we can get through the pentose phosphate pathway. I say mostly because some of it occurs in the mitochondria, but we don't need to worry about that. Because acetyl-CoA can't get through the mitochondrial membranes, we snuck it out in the form of citrate, and then we took it back to acetyl-CoA. Fatty acid breakdown occurs in most tissues and occurs mostly in the mitochondria through beta oxidation. We can't get the fatty acids into the mitochondria as is. Instead, we're going to sneak them in as fatty acylcarnitine through the carnitine shuttles. Lots of sneaking around in this lesson. Also, lots of regulation. The synthesis and breakdown of fats are going to be reciprocally regulated. So if you think about the key regulatory points, when we were synthesizing our fatty acids in the cytosol of our fat and liver cells, remember, the key regulatory point was our acetyl-CoA carboxylase, which the acetyl-CoA carboxylase, remember, it put the carbon group onto acetyl-CoA to give us malonyl-CoA, which was that temporary intermediate that we used in order to hook up the acetyl-CoAs. We had to activate the acetyl-CoAs, and the activation was happening through this ACC. And this ACC protein is going to be the regulatory point of the synthesis. The highly regulated point of the breakdown we just saw was the CPT1, or remember that can also be called CAT1. And that was going to be allowing our mitochondria to take in the fatty acids. We couldn't take them in as fatty acyl-CoAs, but we could take them in as fatty acylcarnitines, and then swap the carnitine for the CoA inside the mitochondria. Let's think about how these are regulated. The ACC, so our synthesis, we see that we have an inhibition arrow coming from palmitoyl-CoA, which is a later on product in this pathway. And so this is going to be an example of feedback inhibition. What about this stimulatory arrow? So I know it's kind of in pink, but don't think that's a stimulatory. There's an arrow there, and it's pink and not red. What this is showing us is that the citrate is activating the ACC. Remember, citrate, this was basically going to be higher up in the pathway because the citrate was how we got the acetyl-CoA equivalent out of the mitochondria because we couldn't take acetyl-CoA out itself. We snuck it out in the form of citrate, and so if we've got a lot of citrate, we want to go and hook it up to form these longer fatty acids, so we want to stimulate the ACC. Feed-forward stimulation. If we are making fatty acids, we're going to be generating malonyl-CoA along the way. And if we're synthesizing fatty acids, we don't want to be breaking down those fatty acids. And so we want to inhibit the breakdown And the regulatory point of our breakdown was going to be that CPT1, that CAT1, which was going to allow us to put the carnitine group onto the fatty acid that would then allow that fatty acid to get into the mitochondria. And so the malonyl-CoA is going to inhibit the CPT1. So there you go, the basics of amino acid and lipid metabolism.